Hello everyone and welcome to Monica's Worm Corner. On today's segment we're going to be talking about GFP tagging. So GFP stands for green fluorescent protein and it was first discovered in jellyfish. So with GFP, a UV light with a blue light range hits GFP, causing GFP to emit a light green light. And so GFP can be used as a reporter gene, and so scientists will use a reporter gene, or GFP, in order to indicate whether their gene of interest has been taken up or expressed in a cell or organism population. And so in our lab we use PCR fusion in order to create DNA constructs. And to better kind of understand what PCR fusion is and what goes into reporter genes, I'm going to use an example with the strain that I used. So I use a strain that, so these worms, they fluoresce green in their body wall muscles. And so what ended up happening is that the scientists who created the strain had to amplify the promoter region of the MYO3 gene. And so MYO3 encodes for myosin. And on the board behind me, I have an image directly from Worm Base about PCR fusion. So what these scientists did is they amplified the gene of interest, which was myosin 3, so they amplified the promoter of MYO3. And they also amplified GFP, and they took those PCR products and they fused them together. And so then they injected it into the worm. And so when you fuse it together, what ends up happening is that when MYO3 is transcribed, GFP will also be transcribed. So the cells that have MYO3 will fluoresce green. And since the body wall muscle has a lot of MYO3 genes, the body wall muscles fluoresce green. And so we can also use fluorescent staining in order to kind of tag biological molecules of interest. And so in my research, an easy way to kind of indicate muscle degeneration over time is phalloidin staining. And the way that phalloidin staining works is that phalloidin will bind onto F-actin, which prevents the depolymerization of actin fibers. And so this phalloidin staining allows for the visualization of F-actin under a microscope. And so we use that to see how the muscle has changed over time as disease progresses. So thanks for tuning in and learning a little bit about GFP, reporter genes, and a little bit about fluorescent staining. I hope you guys enjoyed this segment.